real treat and a pleasure to introduce Dr. David Roberts here as our visiting professor and guest speaker for the Grand Rounds. David's chair of neurosurgery at Dartmouth and a real thought leader in stereotactic functional neurosurgery, image-guided neurosurgery, brain tumor surgery, and many other areas. And David's going to share with us some newer work in fluorescence guidance in this surgery for brain tumors. And I think this is something that is really going to revolutionize how we treat these patients, and really looking forward to your talk. Thank you so much, David.
Good morning. Can everybody hear me okay? All right. I'm going to talk about a project that's been going at Dartmouth for about 10 years that bridges the medical school, the engineering school, arts and sciences, and a number of laboratories across campus. And every year it picks up additional steam. It's an outgrowth of our stereotactic program in image guidance. And that's how we came into it. Maybe it'll become apparent here. It was a means for us to track things at a molecular level rather than just the millimetric co-registration of conventional image guidance. This is the obligatory disclosure, but actually in this instance, it's worth taking a second because this is heavily supported by a number of entities. Number one, Zeiss has provided a fair bit of equipment for us, and we're now heavily collaborating with them. DUSA Pharmaceuticals supplies one of the drugs that we're using, 5-ALA. Medtronics has an industrial partnership with Dartmouth on our image guidance work. And not least important, none of this is FDA approved, and it requires use of an IND that we can talk about later if anyone's interested. George Gabriel Stokes, first baronet, fellow of the Royal Society. He was a professor of mathematics and physics at Cambridge, and he did most of his work in fluid dynamics. The Navier-Stokes equations are named after him, in optics and in mathematical physics. He was secretary and later president of the Royal Society in London. And in 1952, he presented a landmark paper in which he described an observation that there were certain materials, in this instance particularly fluorspar and uranium glass, that could convert invisible ultraviolet light into longer wavelengths that were visible to people, a condition that we now call fluorescence. This phenomenon required the excitation at a molecular level of material that would then, when it returned to its resting state, emit the energy that it had absorbed during excitation. Biologically, there are a very large number of substances that are fluorescent, and some of the most common, NAD, and when I talk about autofluorescence later, they're coming from this. NAD is a large contributor to it. From a practical surgical perspective, collagen and elastin are very fluorescent. Chlorophyll is fluorescent. And protoporphyrin 9, that I'm going to talk about at some length, is a natural molecule that's very fluorescent. We're used to, in our everyday life, our visualization of the world encompasses features like contrast and level. We also perceive color, but we don't perceive different wavelengths. We see wavelengths all as a combined sum. But it's possible that if spectroscopically the world before us has an added dimension that lets you identify things that may look the same in their sum. And in fact, this phenomenon of just spectrally resolving the world around us is an area of extreme research in industry, in smart systems, in systems that have automatic recognition of the world, sorting things in bins. If you do it spectroscopically, you can do it far more accurately than just with the typical vision we're used to. And this phenomenon has been applied in medicine, that the spectral reflectance, this is just white light and taking optical imaging for functional mapping in the operating room, is using this principle here of just reflectance. And if you do that, you can distinguish between tissues that to your eye may look the same, like a malignant portion of a malignant tumor in the brain and the surrounding white matter. If you then incorporate these fluorescent properties, these become much exaggerated, and you can begin to discriminate tissue far more accurately. Neurosurgery has a long experience with fluorescence. 1948, Journal of Neurosurgery, the use of fluorescein in intracranial tumor. And with its administration, tumor would selectively display features that were not seen in the surrounding brain. Fluorescein, and I'm going to show you a number of these. They're kind of fun, but they also, I think, are helpful to appreciate what colors you're perceiving. We'll be excited here, we'll emit here in a green-yellow portion of our spectrum. And it's widely used, it's FDA approved, our ophthalmologic colleagues use it all the time. It distributes in the vascular compartment so that vascular pooling, blood pooling is demonstrated by this. And of course, our high-grade gliomas, blood pool. Also vascular breakdown. This will leak through, and much like gadolinium it will point out features of a tumor for its identification. And you're seeing today a resurgence of interest in fluorescein, in large part because of the FDA restrictions on 5-ALA. It's sort of a poor man's intraoperative fluorescence today. But you're seeing reports where it's being administered during surgery for tumor resection. And even with an unfiltered microscope, there's visible changes in the optical properties here that let you identify tumor. Now, there are limitations that are a reflection of the very properties I spoke of blood pooling and blood-brain barrier breakdown. That's all it can show. So other parts of the brain that share those properties will also take up fluorescein, and it will also leak. It's just an extracellular marker. Now here's another fluorophore that the neurosurgical community is familiar with because of its applications in intracranial vascular procedures. Indocyanine green, ICG, operates in the near infrared, meaning that you can't see its fluorescence. But CCDs that are sensitive at this portion of the spectrum can. And today, operating microscopes commonly will be adapted for ICG so that during a procedure you can administer it and you'll see the blood vessels in the surgical field light up. It's handy if you're clipping an aneurysm and you want to make sure there's not a neck of the aneurysm that your eye is missing. The greatest interest, though, in the clinical neurosciences today concerns 5-aminolevulinic acid, 5-ALA, which is not a fluorophore, but it's a molecule present in all of our bodies in the heme synthesis pathway. The heme synthesis pathway, which is partly in the cytoplasm, partly intramitochondrial, is rate limited by ALA synthetase. And if you administer supraphysiologic doses of 5-ALA in a living organism, you bypass that step, it will race through this channel up to protoporphyrin 9. And there's not enough iron here to go on to perform extra hemoglobin, and protoporphyrin 9 will accumulate in the cell. Protoporphyrin 9, and it will accumulate preferentially in tumor cells. And protoporphyrin 9 is a fluorophore. So 5-ALA converts to protoporphyrin. It sits there, and when illuminated in the violet-blue range, it will glow a reddish color. I don't know, has anybody here, have you used this at all? Has anybody ever seen this? All right, well, non-scientific part. The first time you see that, it's one of these phenomena in the world that the first time you see it, make you go, wow. You know, you can see pictures, you can read all about it, but the presence of protoporphyrin IX fluorescing in the surgical field is like having a glowing charcoal in the surgical field. It just stands out. Now, coming back to this, and this just isn't arcane. There are properties in here that I'm going to actually dwell on in the latter part of this talk. Everybody talks about these wavelengths here absorbing in the violet blue and the 635 peak. It turns out I don't think those are going to be very widely used in the future, but right now they are. The interesting part of this is actually out here, and I'll talk about that. To do this in the operating room, you need two things. It's actually very, very simple, and it's very intuitive. Of all the technologies that are imported into clinical neuroscience, many of them are extremely expensive and very sensitive to having to operate, be very finely tuned. This phenomenon is terribly simple. You administer the patient orally 5-ALA, they drink it in a little cup, about 3 hours before surgery. We do it as soon as they show up in the facility, before they've had any preoperative MRI scans or even gone through the registration process. And then it's being converted to protoporphyrin IX. And then by the time you get in the operating room, it will be fluorescent. The second, number one, you need the drug. And right now, that's the rate-limiting step in the United States. The FDA has not approved it. It's approved pretty much everywhere else in the world. The toxicity profile for 5-ALA, and I'm not going to talk a lot about that, but I'm happy to answer questions at the end, is extremely low. In fact, it's lower than Fluorescein, which is FDA approved, which is the great irony of it. But so to use 5-ALA in this country, the FDA won't accept the German studies, the European studies, the Japanese studies. To use it in this country, you need an IND, which is a lot of paperwork, as I'm sure you're aware. The second thing you need is you need an adapted microscope. You don't technically, you wouldn't really need that. You could just sort of get blue light in there because you can see with your eye the fluorescence I'm going to describe. But today it's generally used with an operating microscope. There are several commercial adaptations that have been made. There's a module that gets plugged into your microscope that does a couple of things. Number one, with a flick of a button, it changes the light source from a broad bandwidth white to this blue-violet. And the second thing it does is it flips a couple filters into the optical path that will highlight and make it easier for your eye to appreciate the fluorescence, although, as I said, you don't really need that. And here we are. You can be operating. You just typically you'll program one of the buttons on the handle, hit the button, and under blue light conditions, the malignant glioma in this instance will glow this pink reddish color. If image guidance was cutting on a dotted line, this is like pink by number. This video just illustrates resection in a high-grade glioma. You can see the image guidance contour around it. During the resection, the tumor bed's very hard to distinguish for the surgeon what might represent malignant tumor. Here, under blue light conditions, it's operating like under a full moon. It's better. It's actually a little bit better than the video portrays. It's possible, although not everyone likes to, it's possible to be operating under this condition rather than just using it as a static image. And you can see portions of the tumor here. And the concept is intuitively terribly simple. If you see the pink, it represents the tumor you're trying to take out. And I'll talk in a second about how it corresponds to MRI features. But the ability to ensure completeness of resection is enhanced by this. And equally important, when you're at a boundary and you're really determined to get it, a surgeon might be more aggressive than he or she needs to be. And this lets you follow a line of resection that enhances the safety. Now, the landmark clinical study is this study by Walter Stummer. It's a multi-center study that came out of Germany, prospective randomized trial, 2006, Lancet Oncology. This is if there's one paper to introduce one to fluorescence guidance for malignant glioma, it would be Stummer's paper. And in this study, there were 300 plus patients who were enrolled in the study, randomized to white light resection or to fluorescence-guided resection. And the study was terminated at interim analysis because it was profoundly positive. That the postoperative MRI confirmation of complete resection was essentially doubled in the fluorescence-guided group. One of the limitations of this study is that a number of centers in the German study were not using the image guidance that is generally used in this country. So it was disadvantaged, and these numbers may be a little lower. 6-month progression-free survival was doubled. The study, although it was stopped, methodologically for all the reasons that had prospectively been in the design of the study, the one shortcoming of doing it right was that it was terminated prior to being powered sufficiently to say anything about survival, which is a real tragedy. I'm going to show a couple other slides from the German multi-center group out of the same pool of patients. If you do a recursive partitioning analysis on the malignant gliomas, and they follow all the characteristics we're used to in terms of survival curves in these Kaplan-Meier's. If you take the RPA class 4 to class 5, and you look at those in whom they were able to get a complete resection, the favorable curve here in all of these instances is statistically significant in favor of the group that had a fluorescence-guided resection. And just for example, if you look at there's not a laser left. If you look at this group here, if you just look at complete resection, the 2-year survival rate, it goes from a little over 2.5% to 11+% by virtue of that. So they're not curing high-grade gliomas, but there is an impact for the patient that's meaningful. Well, what are the risks of this kind of more aggressive resection? As I mentioned, the risks from the drug itself are very low. A significant number of patients will have a transient bump in their liver function tests. Clinical significance of that is rare. They'll be photosensitive for 24 to 48 hours, so most centers will keep them out of bright light. The protoporphyrin IX at the skin will make them susceptible to a sunburn if they were outside. But it's out of the body at 48 hours. The drug's not the problem. The question is a more aggressive surgery dangerous? And the German group looked at this. Turns out that neurologic adverse effects were unchanged between the two groups, with the exception that language deficits were a little greater in the more aggressive resections. And if you looked at NIH stroke scale here, there was an immediate worsening of patients that by 3 months was gone. It was transient. And I think all the surgeons are familiar with this. A more aggressive resection, you're more apt to see this, whether it's from edema or just proximity to eloquent tissue. If you looked at how many required subsequent surgery, this is the fluorescent group here. They had a real impact in, again, keeping that patient at a quality of life for a longer period of time with less intervention. So it's a very, very positive study. And every other study that's been reported with its use now has essentially confirmed these findings. I mentioned we got interested in this coming at it from the image guidance perspective and looking for a marker that would eliminate some of the problems of misregistration later in a case when the brain would have shifted or deformed. And this marker is intracellular. It would move with the brain. So if the brain shifted during surgery, our guidance system at a molecular level would not be affected. But we didn't know what the fluorescence corresponded to, and the German group hadn't at that time looked at that either. So this is a highly spatially co-registered group of patients who, by tracking where we were, we could see what the optical properties of that tissue were during surgery. And you can see in this tumor surgery one instance where there's non-fluorescent tissue, and it's outside the contrast-enhancing portion of this malignant glioma, whereas on the left it's in the middle. If you look at how does it correlate with gadolinium enhancement, I can tell you up front, its properties that make it fluoresce are not one-to-one with, are not dependent upon blood-brain barrier breakdown. It's influenced by it, but not dependent on it. But the correlation, this is the best marker for contrast enhancement, and it turns out the correlation with contrast enhancement is extremely high. And we now believe that the fluorescence extends just a little bit beyond, in most cases, the contrast enhancement of a GBM, for example. If you looked at the grade of a glial tumor, the higher the grade, the higher the fluorescence. I'm going to actually focus on that quite a bit in a few minutes. If you looked at the infiltrating zone of a tumor, these were markers of less than a third, less than two-thirds, between two-thirds and 100% tumor cells. The denser the cell population, the higher the fluorescence. And what did this mean in a glioblastoma group? Sensitivity 0.75, meaning that you were picking up 75% of tumor cells. Specificity 0.71, meaning that if it didn't fluoresce, the likelihood that it wasn't tumor was 71%. But just in the workflow of the operating room, what's really important to us is positive predictive value, negative predictive value, meaning that if you see a cell fluorescing, what's the likelihood that that's a tumor cell? And it's very high, 0.95, meaning very few false positives. But the negative predictive value in this implementation was limiting. It was fairly low. And the reason for that is that high-grade glioma has a lot of necrotic tissue. And all of that necrotic tissue doesn't have the viable cellular mechanism to convert 5-ALA, even if it gets it, to protoporphyrin IX. Here in a case with a heterogeneous MRI appearance, the areas that were contrast enhancing had greater visible fluorescence. And groups doing this, like the German study, had just used subjective visualization of it. I showed you examples where it's floridly fluorescing, but there are many instances, and when people first start using this, it can be very hard if there's very faint fluorescence. You're not quite sure, is this some autofluorescent phenomenon, or is it real? It's very subjective. To get a handle on this, we investigated, because of the tight co-registration, we investigated this in every way that we could. And we had everything from Western blots to genetic analyses. And here on spectroscopy, essentially, the more fluorescent, the more anaplastic its profile by whatever means. This study out of Vienna showed that that portion of a contrast non-enhancing tumor here that was highest on PET studies, highest on some other markers for anaplasia, was the area that fluoresced. And if you looked at things like proliferation indices, they were highest by far here. In fact, twice as high here as in the rest of the tumor. Our interest in this then went into, is it possible to get around the limitations of just the limitations of our retina? Are there ways to objectively analyze that signal that could tell us more? And the short answer is yes. And this was a study where we took the tissue and we processed it for absolute levels of protoporphyrin IX. And you might think, well, the more protoporphyrin IX, the more it's going to fluoresce. And that's true in general, but it's not absolutely true. And the Cliff Notes, again, is that we have many, many instances of tissue with high levels of protoporphyrin IX that do not fluoresce for reasons I'm going to talk about, and others with low levels that fluoresce. So what your eye sees is not terribly accurate. There are other confounding optical properties of tissue, particularly the scattering that you see within tissue and the absorption that limit the emission of fluorescence. When we quantified these tumors ex vivo, and we took grade 1, grade 2, grade 3, grade 4 glial neoplasms, you can see, looking at this, that the profile here, this peak, this is the 635 peak in this instance, is actually discernible even in the low-grade tumors that aren't fluorescent. And this just looking at every parameter that neuropathologically could be looked at, there was a clear difference in the protoporphyrin IX concentrations the more tumor cells were present. And I'm going to come back to this because this is kind of the interesting area. This is ignored in present applications of 5-ALA fluorescence. And you might look at this and say, I left that in high school physics. But you can do a great deal with this. And this information is actually readily available on very inexpensive CCDs like you have in your iPhone. And it's a shame to not take advantage of those properties. So our biomedical engineering program that was part of this optics project just went wild pursuing this. This is a fiber optic probe connected to a laptop that can be placed on the tissue. And in that probe, there are actually 4 fibers that emit either white light, blue light, or they receive the light back. And it's possible using this to interrogate the tissue. Here you can see its use in the operating room. It would be normally used under blue light conditions, but at least in this figure you can see what the probe looks like. And by emitting white light, it's possible to discern the optical properties of absorption and scattering, totally independent of any blue light or fluorescence, and to understand the tissue properties. And if you know those, you can then solve for the absolute protoporphyrin IX levels. And there's where we see the correlation with what's visible. There's a very real correlation, but it's loose and insensitive. When we first tried this out, here was an example, probe on normal tissue, probe on tumor, and you can see picking up, you are now identifying that 635 peak of protoporphyrin IX. And here were 3 tumors that we just piloted this on, and the maximum levels of protoporphyrin IX in normal tissue, you can see here, and the minimum levels within the tumor, you see next to it, on average, 2 orders of magnitude higher levels of protoporphyrin IX. So there's a lot of protoporphyrin IX in there, even though you often cannot see it. All of this is in the context of, while 5-ALA is available around the world, it's being used only for high-grade gliomas, pretty much. It's certainly not being used for low grades. And I think that's going to change. And I'll talk about that. I showed you an ex vivo study of quantitative levels of protoporphyrin IX. These measurements are now in vivo using that probe. And again, a series of tumors, a number of which are not fluorescing visibly, all of which illustrate the spectroscopic signature of 5-ALA, of protoporphyrin IX. In this instance, these are different data points on tumors, all tumors, low-grade, high-grade, meningiomas, mets, and data points that are points during surgery that were tested that were visibly fluorescent are in red. Points that were tested that did not fluoresce to the surgeon's eye are in blue. And this is the control. And this is a log scale. So the level in most of these tumors is exceeding control. This is a receiver operating characteristic curve, ROC curve. It's the metric by which every test we use in medicine, from pregnancy tests to you name it, is assessed. And it's measuring sensitivity against specificity, or in these configurations, 1 minus specificity. If you have a test that has a very, very low threshold to be positive, you'll detect all the tumor cells, but you'll have a lot of false positives. If you had a random test, if you had a fake test, it would be a diagonal line that ran from the origin to the upper right corner. And most tests that we use look something like this. An ideal test would go straight up to 1 and then straight across. There are very few of those. This is an ROC curve for subjective visible fluorescence. This is quantitative fluorescence. There are several ways these are, you can measure these objectively. Area under the curve is perhaps the most common. 0.73 for visible fluorescence, pretty good. But quantitative fluorescence takes it to 0.95. Now, this is a busy slide, but I'll point out a couple of things. This is visible fluorescence and area under the curve on an ROC curve. This is quantitative levels of protoporphyrin 9. And then these are various ways that people have tried to quantify a spectroscopic signature, like the peak of 635 or the area under the curve of 635, but not taking into account the confounding variables of absorption and scattering that make those insensitive. If you look at the area under the curve for high-grade gliomas, you take it up to the 0.95, 0.96 level. If you look at meningiomas, 80% of meningiomas fluoresce visibly. If you measure quantitatively, it's close to 100%. If you look at METs, 0.75, about similar to high-grade gliomas. If you do it quantitatively, it's 0.98. And if you look at low grades, 0.54, which is awful. Quantitatively, 0.75, which isn't anywhere near 0.96. But remember, 0.75 is about what visible fluorescence as used today and as used in the German multi-center study, that's what it was. And I can tell you the technology to do this quantitatively is relatively inexpensive, and it's going to be available to everyone, and it's a game changer. Just like the operating microscope is a game changer for the surgeon, the optical properties of the surgical field that our eyes are limited in appreciating can be greatly enhanced in a way that's meaningful. I think what you've seen so far with 5-ALA is the tip of the iceberg. I want to talk a little bit about low-grade gliomas because people aren't terribly interested in that since so few visibly fluoresce. This is a low-grade glial tumor upon operative exposure, looking at the cortex, blue light, no fluorescence. Image guidance shows quite a ways away from the tumor. And the spectroscopic signature here is typical of autofluorescence, no tumor, no protoporphyrin 9. This is in the middle of the resection. We're focused at a point that's right on the margin. That field is indistinguishable, was indistinguishable to me between, where would that tumor boundary be. Here you can see it really is on the boundary, at least of a T2 signal on MRI. And here's the signature to that probe. And it's very, very strongly specific to protoporphyrin 9. This is our experience with our first 12 low-grade tumors. If you looked at, on this, similar to the earlier table I showed you, the visibly fluorescent are in black circles, the non-visibly fluorescent in white, and all the control tissue here. So its discriminating ability was fairly high, but very small numbers, especially small if you broke them down into subtypes. But nevertheless, the pattern persists across all of these. That's a log scale on the left. So these differences are typically 2, 3 orders of magnitude higher concentrations. If you review the literature on this, this was pulled together a couple weeks ago, for WHO1 tumors, none of them are visibly fluorescent. If you look at a mix of WHO2, everybody's serious, that's done this, it's a couple, maybe a dozen. But if you sum, some of them are visibly fluorescent. I can tell you all of them have elevated levels of protoporphyrin IX. If you look at them, 15% are visibly fluorescent. This is a direction we're headed, and I think as the technology becomes disseminated, this could be a very useful tool, and quite frankly, the impact of complete resection in the low grades will exceed that of high grades. How about meningiomas? I mentioned that 80% of them fluoresce. It's typically a very homogeneous, as you might expect from that enhancement pattern. Very obvious fluorescence, end of resection. Of course, with a meningioma, the surgeons here will say, well, this is ridiculous. I'm not going to leave that kind of meningioma. It's well demarcated from the surrounding brain. But that dural tail, particularly on a skull-based meningioma, it's kind of helpful if you get a marker saying, hey, spend a little more time checking out this area. Here's one where tumor is exposed, meningioma, no visible fluorescence. And then once you're inside the capsule, it's very fluorescent. Here's one, capsule's not fluorescent, and there's no protoporphyrin IX in it. Here we are, we're in the tumor, no fluorescence, and there's a very, very strong PP9 signature for tumor. And if you look at those curves, very similar enhancement on an ROC curve for meningiomas. Now, I've been talking all about tumor, but 5-ALA will also be processed in non-neoplastic tissues some of the time. Here's a rat model, pilocarpine rat model that's used a lot at Dartmouth. And since gliosis will oftentimes process 5-ALA into protoporphyrin IX, we looked at it. And piriform cortex layer 2, where you would expect it in an epileptic animal, strongly fluorescent. Could it be a guidance tool in this application? Don't know yet. Here we are in the hippocampus, CA1, fluorescing. And in fact, I think the non-oncologic applications in the operating room of optical guidance are going to be widespread in the near future, whether it's for epilepsy or any other kind of application, because fluorophores, and I'm going to end actually talking about this, can be used to label other targeting molecules administered to the patient, molecules that can identify epileptic zones, that can identify normal tissue. If you want to identify a pathway that has any molecular distinguishing features in it, you can use this to label normal structures. There's work being done outside of neurosurgery to identify ureters, to identify peripheral nerves, and so forth. You can have these markers that will fluoresce. Well, what are its limitations? Problem is, the concept is you see fluorescence, you remove it to get a complete resection, but you don't know what's deep to the surface. 5-ALA and protoporphyrin IX are visible only to the surgeon on the exposed surface, the veneer. And if you have blood, if you have gliosis, if you have intervening non-fluorescent tissue, it will look like that. And you don't know, is there tumor that I intend to resect that this isn't helping me with? Well, there are ways to get around it, and we're very interested in this. Here's the problem. This blue light doesn't penetrate. tissue very well at all. And certainly anything being admitted from deep isn't coming out. And here this is a CNS1 brain tumor model in rat, has an embedded tumor, white light image, conventional fluorescence, no fluorescence. But if you excite on this portion of the out here, you can see at depth. And there are a couple of problems here. I've shown in that right-hand graph the absorption curve of hemoglobin. Another example why non-quantitative fluorescence is limited at surgery. It's right at the level where we're exciting in conventional applications at the violet blue. It's very inefficient. This part of the curve out here, separate from that 635 peak everybody's familiar with, is in the near infrared. You can't see it, but it penetrates tissue extremely well. And the advantages of that penetration far exceed the lower absolute level here. And our interest is all now utilizing this portion of the signature. And if you take two of these peaks out here, and you look at how much they are attenuated traversing intervening tissue, it varies with the frequency. And by taking the ratio of either the emissions coming out here, you can actually determine how deep the fluorescence is. It's kind of cool. Here in phantom work, for example, we're exciting at different wavelengths, and we can measure the true depth of that rat tumor or the phantom tumor. I think you're going to see more of this. We're currently seeing to depths of about 10 millimeters, which is a scale that would be very, very useful surgically. Now, how about tagging these? Let's get into the really interesting area. You can take fluorophores, particularly in the near infrared, that has the ability of it's not confounded by autofluorescence at all. It has the disadvantage that you can't see it, so you need a camera system just like we do for ICG and vascular applications. But near infrared dyes are really hot in development right now for this purpose. You can tag them with put a targeting molecule that can be an antibody, it can be a nanoparticle that will you can identify receptors, whatever you want to look at, and put one of these on and have it fluoresce at a molecular level. And this is just from a review paper that was just looking at a whole slew of potential targeting molecules, and here are ones that are currently in the preclinical stage. A couple of these are actually now in clinical outside of neurosurgery. And looking at these ligands here, the molecular targets and their applications, our general surgery colleagues are interested in this in breast. It's being used in liver. It's being used in colon. And here's one that we're developing that's looking at EGFR in glial tumors. It turns out that you can identify and have only those tumor cells that express, in this instance, that have EGFR on them. They're what will fluoresce in the surgical field. These will distribute not just in the vascular compartment, not just in the cells that have processed to a fluorophore like 5-ALA going to protoporphyrin 9, but they actually here have an affinity for particular cells. And if you give, in this instance, two dyes at the same time, one of which is targeted to the cells and one of which just distributes the way Fluorescein does, you can actually tease out what these rate constants are. And you can quantitatively know what the molecular density of the targeted area of interest is. You can calculate the binding potential and the density of those receptors. Is that going to be useful in neurosurgical oncology? I don't know, but it's tantalizing, and I think it's very possible that it could be. For the residents that are here, this is again the tip of the iceberg, and I think there are tremendous opportunities in development here. It's a frontier today. These near-infrared dyes, very few of them are currently FDA approved, but I'm certain in the next 5 years that there will be a number that are available. And all of the work and all of the potential applications of these in neuro-oncology remain to be discovered and exploited, and I think it's a great career opportunity for someone interested in neuro-oncology. Here's one that's interesting. Activatable probes. These are fluorophores that don't fluoresce until they've been activated. That have been activated in this instance by some molecular marker. And a good example are those that take place of matrix metalloproteinases. These are present in glial tumors. They're responsible for the infiltration and migration of malignant glioma. They're not present at any significant level elsewhere in the brain. If you administer a fluorophore that distributes widely but doesn't become fluorescent until it encounters these MMPs, then you could have a marker that might be more specific, or at least would be targeting a different moiety than 5LA would. And here's some mouse work that we've done. In this case, this is a U251 tumor. White light image, raw fluorescence here, quantitative fluorescence here, and then superimposed on a heads-up display, illustrating where this metalloprotase activated fluorophore is distributed. Here's another one, same thing. Very powerful, very, very easy to use, very user-friendly. And just it's correlating with the histology. Now, you might say, well, wait a second, that probe, all that's fine, but taking that probe and going around a surgical field holding that probe, interrogating these different points, sounds awfully inefficient. And it would be. It takes very little time for that probe. We collect that data in literally seconds, but it processes over the course of less than a minute to come out. But if you have thousands of points in your surgical field, you're not going to want to do that very often, or certainly won't do it across the whole field. Is there any way you can get around that problem? And the answer is yes. Here's an operating microscope that's been modified with a liquid crystal tunable filter. That's a little device that over a matter of a few seconds changes through all of at each frequency optically across the field and collects all of that data on that camera. And it does it, we do it in white light, and then we do it in blue light. And each of these are different settings of that filter so that you can get the spectroscopic properties of each pixel in the surgical field. And you can do it, when you do it under white light, we do that specifically in this application looking for the absorption and scattering optical properties of the tissue, that if you know that, you can get the quantitative levels of protoporphyrin IX. And then we acquire a blue light in the same way, and here you can see that 635p. Under white light conditions, you can see you get a stack of images here, one for each frequency bin, and every pixel in that stack, represented by a line here, gives you the level of that emission so that you can generate that curve, and in turn calculate the absorption and scattering on the right. Now, when you do that under fluorescent conditions, you similarly get that typical curve that you're all now very familiar with. And when you put those things together in the surgical field, you can get for every pixel a breakdown of all of these properties. And I've been talking about protoporphyrin IX, but it turns out that the same spectral resolving lets you look at any of the fluorescent moieties that are in that pixel. We can look at other products besides protoporphyrin IX here. There are actually other lesser or lower levels of other fluorophores in the field, but there are other fluorophores, just like oxyhemoglobin, deoxyhemoglobin that you can also get so that you can use this for functional imaging. Kind of nice if you are doing a wake cranie, if you want to see, this is an area that we're greatly interested in right now, language mapping, wide field, across the field. Okay, that's theory. Here's an application. Here's the liquid crystal tunable filter. Here's the camera adapted onto the operating microscope. And here was our work in Phantom, illustrating what you want to see here is the linear correlation between the actual, in this instance, protoporphyrin IX level and the ability of this camera to quantify at each pixel those levels. This is very usable. Actually, I'll skip over that. And here is an animal tumor model, white light conditions, upper left, blue light conditions here. You can see some of the faint fluorescence here. Here is the quantitative map, so that every pixel here, we actually know the level of protoporphyrin IX, and here it is in our heads-up display. In this instance, here's, again, tumor model. This is a CNS1 brain tumor model, white light conditions. This animal has been administered 2 fluorophores, Fluorescein and protoporphyrin IX. And to your eye, the Fluorescein emission swamps out any other fluorophore, and you just see this green-yellow image. But if you look at 1 pixel here, you will see in that the signature of Fluorescein, the signature of protoporphyrin IX, and this, of course, is your total summed picture, but this information here can be quantitatively teased out. If you look at another area here, it turns out there's very little Fluorescein there, but a fair amount of protoporphyrin IX. And this kind of system can give you a quantitative map for each of those fluorophores. In this instance, protoporphyrin IX, you can see a map very similar to the map you would see if that were the only fluorophore that had been administered to the animal. And here's a quantitative map for Fluorescein. And of course, they're very similar, but they're not 100% concordant because they're showing different properties of that tumor. Will that be useful clinically? Don't know. Maybe one of you will find out for us. Okay, now let's take it into the clinical arena. Here is a patient undergoing GBM resection. White light conditions, blue light conditions. This tumor really came up subpelally so that you can actually visibly see some of the fluorescence. And here's a quantitative map. During the resection, near the end of the resection, think you're done? Okay, let's use this in the way that it's commonly being used. Blue light conditions, is there any tumor left? No. But what about at this mark here? There's a signature. Here's our enhanced, superimposed, quantitative level of protoporphyrin IX, and there is tumor. And this is wide field. So we've gotten around that probe problem for this application, and we've now used this in 12 GBM cases. Further resection. So I will leave you with the following thoughts, that although fluorescence has been around for a long time, the current resurgence of interest in 5-ALA has, I think, gotten a lot of people looking at other fluorophores, other technologies that can enhance optical imaging. When we're in the operating room, the operating microscope is just a fantastic instrument, as all the surgeons here know. The illumination is great, the optics are great. But it's a platform that we're not using an awful lot of information that's accessible during the surgery. We throw it away or we don't perceive it. The ability to extract more information from that surgical field in a way that is very efficient. You don't have to stop the case to do an MRI scan for an hour. It's real time. It's there. It's very easy to use. It's quite inexpensive compared to these other technologies. And it's like the calculator that used to be on our wristwatches. Most people never used them, but if you wanted them, you could get a lot of computational power that might be useful. There's a great deal of development going on today looking at other fluorophores. I've given some examples today. And this use of targeted agents I think it's analogous to what's happened in the chemotherapy world, now being brought into the operating room. And the computational power required to do these things is every year less and less expensive and accessible. So that I think this is a technology that has a great future in neurosurgery. This work is that of a large number of individuals. I particularly single out Keith Paulson, my longstanding collaborator in our biomedical engineering program at Dartmouth, Brian Pogue, who's headed up the EGFR fluorophore development at Dartmouth. Pablo Valdez is an MD PhD student at Dartmouth who's made a project of this for the last 5 years and done a lot of the heavy lifting. Brian Wilson at the University of Toronto is a giant in optics. And Brent Harris, one of our neuropathologists who left, has remained with the program. So thank you. And with that, I'll open it to questions.
How stable are some of these agents? Because as you keep shining light at them, I know from Evans Blue, for example, it's rather evanescent.
Right. They all will bleach under when illuminated. At a practical level, it's a non-issue. For example, its use in high-grade gliomas, you can have that light on. We'll have, a dozen or a half dozen visitors in the OR, all coming to look under the microscope with it still on the field. And over the course of many minutes, it doesn't change its perceptible occurrence. Even more than that, though, when you're using this to resect, you're constantly working. I'll typically have an ultrasonic aspirator in, and you're constantly removing the exposed level of tissue. So even if you had a fluorophore that would bleach out in a matter of a few seconds, it would still wouldn't be a huge issue.
You protected it at depth.
Yeah. Yeah.
There's an exciting glimpse into the future. And I wonder, in the United States, what are the efforts, or are there efforts to get FDA approval for 5-ALA?
It makes solving glioblastoma easy. It is such a quagmire. I used to, I had been led to believe 5 years ago that, any month now your approval will go through. And I think the FDA is full of a lot of very bright, well-intentioned, hardworking people who do a real service. I think they are so over their head. I think they are so overwhelmed with the flood of what comes before them that this process has been mired in it. And I don't know whether it's going to be approved next month or ever. When the data comes in, as I alluded to, the safety profile of 5-ALA is so benign, and yet they've had it on their plate for 5 years and nothing's moved. Maybe I'm being a little hard on the FDA. There's some other circumstances that are also responsible. Most drugs get through the FDA because industry is sponsoring them going through, and there's no sponsor for 5-ALA. 5-ALA costs pennies. Your chemistry department here has it and/or can make it very easily. So the licensing rights for a company to have it, the German company that has it, hasn't pushed it. Two of the operating microscope companies that are prominent in the neurosurgical world are selling these modules for about $40K, and they're selling. They're saying, "Hey, we're just an equipment company. The drug is somebody else's problem." So they're not sponsoring it, and nobody's pushed it through the FDA to collect the data and everything that's needed. There's been a concerted effort, consortium in this country that has been trying to get this through the FDA now for 5+ years, and it's gone repeated. We've gone before them multiple times. We took a prospective trial to them about 3 years ago, and it got so mired down in the, I shouldn't say politics, but with the well-intentioned regulatory environment that it essentially ground it to a halt. They ended up, for example, its utility to accomplish a better resection. I didn't show before and after. Maybe I should have slides. My oncologist can tell when we've used it and not used it. If the intent is to resect that tumor, the resections come out like you just lifted the contrast-enhancing tumor out of the brain with nothing extra and without residual. They can spot it like that. And if you say, I want to approve it as a diagnostic tool in the OR to facilitate what we're doing, the data is unequivocal. I mean, the German study alone nails that one, but everybody's replicated that. But the FDA comes back and they say, no, we want survival data. Well, survival data in GBM surgery is so confounded, and it's not just all the variables of age and location. It's dependent on the surgeon. There are so many things that you need thousands of patients to run that study, and nobody is willing to put up the amount of money required to run a study like that. So I don't know what's going to happen. David Thomas, who I'm sure some of you know in London, I was on a bus with him maybe 5 years ago, and I said, boy, how lucky that you have 5-ALA and you can use it. He said, I don't use it. I said, what? He said, it's wonderful. It's easy to use. It really helps me. It costs thousands of pounds in the cost of the drug, and my institution won't let me use it. And if industry decides that they'll make it available, but they're going to charge thousands of dollars, then I don't know how often it will be used. I mean, God forbid if anybody in this room has, or a family member has, a malignant glioma, I can tell you, go to some place that has 5-ALA because the resection will be better. I firmly believe that. Yes?
I was just wondering if you tried in the lab or the clinic, like a multi-photon approach to get at, deeper tissues.
Yeah.
Less organ plasticity, that sort of stuff.
Yeah. There's a lab program at Dartmouth looking at that. Brian Pogue has done a lot of work with different imaging modalities exploring that, but it's a ways from, at least in brain, seeing clinical use. The number of ways that you can, the number of optical techniques that can be, this is scratching the surface. And, Raman spectroscopy, for example, powerful tool, and we're looking to incorporate that. I think it's. I would just also add again for the residents or students here. I remember when I was in training, you hear stuff, you see stuff, and say, well, I'll finish my residency, I'll secure a position, and then I'll get to work. And I can tell you that's not right now. A medical student who may be sitting here, here's an opportunity to run with. That this is right at the beginning of something that's going to explode in medicine and in surgery, and how to use it wisely, what other ideas you have. The ideas that come from a medical student or a junior resident are oftentimes better ideas that are coming from the most senior people in a department. And those ideas that you may have, in the shower or on the weekend, run with them, develop them. Pablo, Pablo was a second-year med student, and he has just spent the last 5-6 years of his life running with this, and every area that you develop is something new that people haven't done before, and yes, many of them are dead ends, but some of them are going to stick. And I think it's a tremendous opportunity.
On that note, thank you, David.